Yeah, thank you so much for inviting me to come today. It's been a very interesting day. I think a lot of really strong talks. I was pretty excited to see all these ones about the coronavirus and that we have some potential ideas about how we might be able to target it. I'm going to be talking today actually about some capabilities that I've built at Biogen in my lab about using CRISPR-Cas9 functional genomic screens as part of our small molecule target identification efforts. So I run a chemical biology lab. Oh, here we go. That is focused on trying to discover small molecule targets of action. So primarily arising from phenotypic screens, because one of the big challenges in phenotypic screens, I mean, there's many challenges, but one of the biggest is actually understanding what the target might be and whether that's a target you're interested in and actually whether it has safety liabilities and other such things. So from a high level, the agenda today, I'm going to talk about just an introduction briefly to what compound sensitized CRISPR-Cas9 screening is, then talk about a phenotypic screen that's been run internally at Biogen looking for small molecule modulators of ASO uptake, and then discuss an arrayed full genome-wide screen that we actually did on our hit compound to try to understand its MOA. So as I was saying, we're really interested in trying to understand what our small molecules are targeting. And there's a lot of different tools out there. One that my lab uses a lot is chemoproteomics. And that's something that's a real workhorse for us. But that's not always applicable to all situations, especially when you can't make a good probe, a good photoaffinity probe with sufficient affinity and efficacy. We use a lot of things like knowledge-based approaches and cellular profiling approaches. But what I'm going to talk about today is the CRISPR approach, which has one big advantage is you don't have to do anything to your molecule. You can use exactly the molecule as you found it from your phenotypic screen, and you don't necessarily have to have a super potent molecule as long as you can see your phenotype. And I think everyone here today online on the call is pretty familiar with CRISPR-Cas9. So I'm not going to go into how it makes its edits, but more how we harness it as part of our screening technology. So when the CRISPR-Cas9 makes a site-specific DNA break, there are two main repair mechanisms: homology-directed repair and non-homologous end joining. And most of the time, the cell actually uses non-homologous end joining. And this process is error-prone, which creates gene disruptions by insertions or deletions, otherwise known as indels. So for the screen I'll talk today, we take advantage of this pathway to actually create specific gene knockouts across the entire genome to try to understand our compound's MOA. And we also have the ability in the lab to do what are called CRISPR inactivation and CRISPR activation screens, which use a dead Cas9 enzyme to actually turn on and off the transcription of the gene. But I'm not going to talk about those today. So what exactly is a compound sensitized CRISPR screen? I mean, what we want to do is try to identify genes related to the action of our drug, as well as identify some genes that might modulate the sensitivity. And so this cartoon is really the simplest example of a compound sensitized CRISPR screen. Really, the original papers in 2016 were these sorts of screens that are really survival screens, either in oncology space or antiviral space, where you have a cell that in the presence of the drug dies. And what we're just doing is we're asking the question, if I knock out each gene individually, does that do anything to my phenotype? In this case, does my cell still die? Well, then it's probably not related to the action of my drug. But if my cell survives, then that gene is probably in some way related to how the drug acts. Now at Biogen, we pretty much never do survival screens because we're in neuroscience. We're 100% invested in the neuroscience space. And so generally we want cells to survive or behave better, not to die. So we don't really do survival screens, but it's a nice just illustration of the simplest type of screen that you can do. And there's two main types of CRISPR-Cas9 screen formats that you can use. You can do a pooled library screen, shown here at the top. And both of these techniques, by the way, have their pros and cons, and I'll get into that briefly right now. So in a pooled library screen, you're going to have your library just in a single tube, so that's pretty cheap to make, relatively speaking. You're going to infect your cells at a low MOI so that you don't have more than one guide per cell, and you're just going to grow these cells up in a single vessel. So again, this is fairly cheap and easy to do from a lab supply point of view. You're now going to do a positive or negative selection for your phenotype of interest, such as survival, or for example, a FACS sorting based on a fluorescent marker. And you're then going to have to do sequencing to understand exactly what guides are enriched in your sample, and then do that statistical analysis. So again, cheap and fast, but it tends to be a fairly blunt instrument. It's going to be, the cells live or die, or do I phosphorylate a protein or not? Fairly simple readout. Now, an arrayed library screen, in contrast, every single well is going to have its own gene targeted, usually by multiple guides to that gene. The screen that we do has 4 guides per well, but a single gene at a time. And then you're going to transduce or transfect in your cells, do your treatment, and now you're going to be able to look at phenotypes. So you can see right away, one of the advantages is you're going to be able to look at every single gene knockout. You're not going to have things that you just lose because they're no longer represented, because you have some sort of it has like a toxic function of knocking down the gene, but you also can look at things much more complex, cell morphology, and in the case that we're looking at, we can look at both cell survival and our actual reporter gene phenotype. The disadvantage of this sort of approach, of course, is that it's going to be more expensive. You have to buy these arrayed libraries, and in-house we have a knockout library and an activation library. And you also have to have the automation and the robotics and whatnot to be able to handle the larger amount of plates and complexity of, we usually do a screen on about 50 plates, 5,384 row plates, which is a lot more complicated than growing your cells in a single vessel. So now I'm going to switch gears and just talk about our phenotypic screen and the problem that we are trying to address now with this CRISPR platform. So Biogen has one of the first antisense oligonucleotides on the market, and that's Spinraza for the treatment of SMA. And these ASOs, the reason that they can even get into cells very effectively is that they have a lot of chemical modifications to the bases and the backbones that allow free uptake. However, a large proportion of these ASOs never actually engage with their genetic target because they become trapped in the lysosome. Oh, I'm sorry, that jumped ahead when I tried to use the mouse. So as the ASO is brought into the cell, they get trapped here and never actually released to do their function. And we call that non-productive processing. So we're actually interested in running a high throughput phenotypic screen to look for small molecules that might actually improve this uptake and efficacy. At the same time, we hope that we'd actually gain a better understanding of how ASOs are processed in this uptake process. So now I'm just going to briefly describe the screen that we use. This is a reporter gene assay. And this is one of course, the biggest problems based on the talk that you saw earlier from Tim, is that this is an engineered cell with a reporter. So it is fairly distant from what we'd be actually wanting to see in a neuron. But we do have secondary assays to do the follow-up. And this reporter is looking at a fairly universal mechanism of splicing. But it is one of the big caveats of the screen. And when I talk about our follow-up at the end, I'll come back to this point. So this is a mini gene reporter where it's using actually part of the SMN2 gene, exon 6, 7, and 8. In SMN2, exon 7 is usually skipped. So in the absence of ASO, that exon is skipped, and there's a native stop codon read in exon 8 that causes the transcript to stop at this point, and you don't see any GFP expression. In the presence of ASO, that exon is now included, which allows you to have the read through of the gene and expression of GFP. And so here on the right, you can see that in no ASO, we have a low amount of GFP signal, and with ASO, we do see increase in signal. Now, I want to make this point that the GFP signal is present in the absence of ASO, and that's because there is a basal level of exon 7 inclusion even in the absence of ASO. And that also is true for SMA patients who rely on the SMN2 gene to produce their SMN protein. So we'll come back to this because we have some false positives in our screen that actually were pretty easy to triage out. Was because there is this basal level of GFP expression. At the same time, we also have a reporter that reads out mCherry with a nuclear localization signal, and that allows us to look at the health. So we're really reading two phenotypes at the same time. Again, one reason that the arrayed screening is a big advantage for us here is that we can actually go back and look at both of these phenotypes. So the screen was run. This was led by Patrick Falloon and the high throughput group at Biogen. And shown here on the right is with 5 micromolar ASO, you have a reasonable amount of GFP expression. But in the presence of the compound, you see that a greatly increased amount of GFP without a big effect at all on our nuclear M-cherry signal. So our cells have stayed pretty healthy during this process. Shown here on the left is just a concentration response showing the ASO alone in blue. And then as you titrate in compound, you get a shift to the left. The EC50 becomes more potent for the ASO, and this happens in a concentration responsive manner. As you increase the amount of compound, you see a further left shift in that curve. Now, we couldn't do chemical proteomics. We've made probes of this compound, but they were never very potent. And actually, the SAR in this compound is pretty tight, so it's hard to get a much more potent compound than 1.5 micromolar. So it was a really good place for actually us to apply our CRISPR screening approach. So this is just an outline of our CRISPR screen. And I'm not really going to spend a lot of time in all of the optimization that we did, though I will in the next few slides talk a little bit about how we evaluated our library and our Cas9 clones. But the basic idea of this screen is that we're going to array our library in our plates, and this is a genome-wide. This is just over 19,000 different genes targeted by the library. There's 4 guides per well. We're then going to transfect by plating the cells onto our guide with transfection reagent. And we've optimized this that 72 hours is the optimal amount of time for the Cas9 transfection and knockdown. Then we treat the cells either with just ASO or with ASO and compound. And this is important because we're actually getting 2 readouts. We should be able to understand genes that are related to our compound MOA, but also identify genes that are just related to ASO uptake and efficacy in general. 48 hours later, we read for our GFP signal, and then that's how we're going to determine which guides are having the most effect. But we also are able to do a genomic DNA QC, which is something that we had to develop in the lab because we didn't want to just rely on a few genes to say that our system was working. We wanted to be able to look across our library. And this was something that wasn't really out there in the literature at the time. So what we do here is actually a pooled indel analysis. Prior to this, we were doing sort of standard Sanger sequencing, tied analysis to look at gene knockdown efficiency. And we wanted something that would be much higher throughput. So what we actually developed was this Nextera NGS approach, where we have amplicons. These need to be 1 to 2 kilobases so that they can work with this tagmentation approach. Sometimes these amplicons contain 2 guides. Sometimes they just contain a single guide each. We extract DNA for each well and do a multiplex PCR. And then we take that into the Nextera NGS tagmentation approach. And what this does is it uses a TN5 transposase, which is able to cut as well as install sequencing adapters at the same time. And then you can use a limited PCR using index adapters to actually put individual index codes for every well from your screen. So every gene will then have its own index, which you can then pool and analyze by sequencing. And this is just showing PCR sequences as a reference. This is an example where you have a single guide and you can see the indel there. And this is actually an example where we had 2 guides and we actually saw disruption between them. And we use this to calculate our indel rate for each gene. And so as part of our assay optimization and development, we looked at 42 genes and asked the question, do we need to have a clone or can we do a Cas9 pool? We also wanted to know, could we use this technique with fixed versus live cells? Because when we do the imaging, we fix, but then we'd actually like to be able to come back and look at, we look at about 2%, 2.5% of our library and ask the question, how well did our screen work across those 500 genes? But then we needed to work with fixed clones. And so what's being shown here on this graph is on the x-axis are the gene names. On the y-axis is a calculated indel rate. So each dot represents an amplicon, and then each bar is actually a calculated indel rate, which is the probability of the cells having at least 1 indel at the targeted gene. And there are some assumptions here. We assume that the guides act independently, that they all get into the cell and act independently. But we use this to quickly ascertain what our best clone was, as well as to QC our actual screen. So you can see here on the left that the live Cas9 pool samples show not as good editing as what we see when we go clonal. This is always a question to ask, do you have to go clonal or not? With this cell line, we really needed to go clonal in order to get our best editing efficiency. And these few knockouts here are really probably, we actually think, failures of PCR, not complete failures of editing. When you go to the fixed cells, you do get a lower multiplex PCR efficiency, so we underestimate our indel rate. However, we did use this as part of our full screen where we looked at about 500 genes, and we saw even with this loss from the fixed cells, we had about 75% of our genes were showing a greater than a 75% knockdown. So we felt that our screen actually worked pretty well. Before I get into the actual data, I just want to talk about what sort of data we expect to see, because it is a little complicated. So first, we're looking at genes related to ASO efficacy by itself. So this is just ASO plus DMSO. Most genes are actually going to look like wild type. So genes A and B, they don't affect ASO activity, so they look like wild type. Gene C, which normally helps ASO uptake or activity, when you knock it down, you'll see a decrease in signal. And then for genes D and E, you see an increase because those usually repress ASO activity. Then once we do this in the presence of compound, we have here genes B and E, which interact with compound. And what you see is that for gene B, which normally has no effect, when you have compound, when you knock it down in the presence of compound, you actually see a decrease in signal, shown here with this bracket. And then conversely, when you have a gene like E, which normally blocks ASO activity, the compound interaction is basically mimicking that. And so you see a lowering of the difference between the two, as shown here on the right. So that's the sort of data that we expect to see. Part of the complexity here, though, is if you have 19,000 genes, it's really hard to look at a graph like this. So we generally graph it as a scatter plot where we put our ASO plus compound on the x-axis and ASO plus DMSO on the y-axis. And these are the exact same genes with the exact same lettering. Most of your genes are going to fall here in the wild type. Genes C and D, which don't involve compound, will actually be here on the diagonals. And then the compound-related genes, B and E, will actually fall on the X and the Y axis, respectively. So with that, I'm going to now show you the data from our screen. So the blue dots here, as we said, most genes show no effect when you knock them down. That's just the ASO plus DMSO. And the red is the ASO plus compound. And then we ended up using the 6 median average deviation instead of standard deviation. And that was because we actually didn't want too many hits to actually look at. So these are the regions of our potential hits. And you'll notice this is an alphabetical display of genes. You do notice this column here, which works both with the compound and without the compound. This actually turns out to be a false positive based on that basal GFP expression, which I'll be able to show you a bit later. But what you can tell is that all the proteins in that particular family were hits in our screen. So now here's the easier to look at scatter plot of our gene hits. And you can see that most of them are falling into this region of general ASO uptake, but we do have some that are in this region of compound-related activity that we're specifically interested in, where it looks like it might be generally activating compound activity, activating ASO activity, excuse me. And so we looked at these genes for follow-up and picked about 700 genes that we then took into our confirmation screen. The confirmation screen we did with more replicates. Our primary screen was 3 replicates. Our confirmation screen was 6 replicates, and we used a more stringent test. But there's one other thing that we did that was important, is we did this no ASO control. And as I told you before, there's this little bit of basal expression, and it turned out that we knew from running a set of bioactive compounds that things like protease inhibitors gave a false signal in our assay, presumably because the GFP protein, that little bit of basal expression of that GFP protein is not being broken down, and so it's building up and giving a positive signal. So if you look here now at our confirmed hits, about half of our hits actually phenotypically confirmed, and about a little under 2/3 of them actually were dependent on the ASO. But the ones that are ASO independent are all these purple X's. So a lot of our very strong hits were actually false positives from the ASO independent mechanism. And when we actually go in and look at these are just some of the numbers of the ones that we know are confirmed hits, including the 7 that are actually the ones who are most interested in following up originally. But just really quickly, I wanted to say that the false positive genes were really what we expected them to be. They mainly hit pathways like ribosome, proteasome, spliceosome, and RNA transport. And we were able to very quickly triage those out. Now, I do wish I could actually tell you the genes that were our hits of interest, but we're still following up on these, and we haven't published yet, so I can just give you a little bit of a teaser. We had 7 genes that were truly seemed to be pathway specific for the compound, and of these 7, all of which are interesting, but there were these 3 that actually were tightly related. A and B are actually part of the same complex, and then C was known, well documented in the literature, to have direct interactions with this pathway. And in this graph here on the right, I've actually colored the confirmed hits related to this pathway in red, and you can see that there's actually hits over here in the general ASO region, including, very interesting, an E3 ligase for gene A, which kind of makes sense, right? Because what we're saying is that if you knock down gene A, you have less response to compound, but that normally an E3 ligase is effectively blocking and knocking down gene A. So if you knock down the E3 ligase, you should actually see presumably more gene A, which would actually give you an increase in ASO signal, which is exactly what we see. So we're actually pretty excited in this pathway. We're also looking at some of these other just black dots that were not related to the compound or that pathway. And some of these are related to early endosome and the Golgi and autophagy pathways. So we're actually pretty interested that these, to further explore what these might mean for the ASO uptake mechanism. And because we are in a pretty artificial system, the thing we were working on right before the COVID-19 shutdown was actually validating these in other cell lines, to ask the question, just to make sure it's not some artifact of the cell that we're using, but is this a mechanism that will work in uptake in other cell lines? So we're looking at a sushi cell line, we're looking at neurons, and other things like that. So I think that's about what I had to present today. We've actually built a pretty robust screening capabilities at Biogen in the last 2 years, as well as this novel QC method that allows us to look at indels of 500 genes at one time. With our full genome array CRISPR-Cas9 screen, we have found a common pathway related to compound mechanism and are following up on these genes. And we were able to triage out our non-ASO related genes very easily. And I didn't have time to go into it today, but we are building capabilities to actually perform these CRISPR-I and A screens in iPSC-derived neurons, because that's ultimately where we're going to want to go, so that we can get away from these artificial cells and try to get into the cells closer to the patients and the biology of interest. And with that, I just want to thank the people involved, particularly Patrick Falloon, who was the project lead for the phenotypic screen, and Yiying Chow, who came to my group just under 2 years ago and built these capabilities at Biogen. And with that, I'll take any questions.
What a wonderful talk. Thank you. It was a very beautiful talk. I mean, that's ultimately the future of systems biology, what one can see here, because it really, you are able to capture the entire system response. That's really, I think, what's needed. So congratulations for very nice work.
Thank you.
So, do we have any more questions from the...
I have one question, if it's okay. This is Per Greisen. Very wonderful talk that you did there. I was wondering, now that you saw all of these false positives, if you actually consider to go back, because there must be a lot of false negatives in your screen as well. or what is your feeling about that?
I mean, there probably are some false negatives. We ran, so one of the things about CRISPR screens that are kind of nice is that you tend to pick up pathways. And so we had statistical tests for making our cutoffs, though there was, something in the pathway very related to those 3 genes I talked about, and it just barely missed significance, and could have been selected actually if we had used a different statistical test. So because you're kind of looking at pathways instead of just a single target, you're able to go back and say, oh, that was awfully close, and now I can follow up on it. All right. But sure, there are false negatives. One of the reasons, though, here that in particular that we had the false positives is that the splicing mechanism already we knew had this exon 7 inclusion just a little bit, and we already knew from our chemical genetics set from compound libraries that we were going to get these false positives going in. So it was no big surprise, and we were prepared. We knew that our first thing we were going to do was a counter screen to look for it without ASO.
Makes sense. Just a comment to that. So you say that the pathways make sense, but in principle you could have orthogonal pathways that you actually would not pick up in the false negative ones, right?
It is possible. I think the chances are you'd find something, right? We tend to sometimes find 3 out of 4 subunits. Well, that 4th subunit, maybe the knockdown didn't work well, right? But we hit other things that were related. If you're going to like say that none of the knockdowns worked and you missed them all, like the odds are kind of in your favor that you'll pick up.
No, I get that.
Of course, there's always a chance you might miss something. But we had a situation of one of the genes we're interested in following up on where we found 3 of 4 subunits. So we're pretty confident that 4th subunit is some false negative, right? And that we can go back. And that actually went along with our QC. When we looked at those 500 genes, we had about a really good editing of 75% of them for sure. So we're like, okay, so there's probably 25% that are partially edited or maybe not edited at all. And so we'd be able to catch those. Yeah. So it does take a lot. There are resources for follow-up.
How did you actually pick out the 700?
So that was done, so for that we ran so when we did the later statistical test, it was a Dunnett's test, which was pretty rigid, so strict. The earlier one, I'm trying to remember what statistical test we ran. It was a little bit more loose, and a Wilcoxon, so it was a little bit more generous. And we looked by quadrant. So for example, if we're compound related, we only wanted things that were compound specific. And so if they were specific for ASO without compound, we actually ruled them out. But doesn't mean they didn't have an effect. We just ruled them out based on those statistical cuts. And we wanted to be under 1,000 total because that's what we felt we could buy in order to do the follow-up. So we got 700 sort of as a financial balance of like how many hits do we think we can handle. But you can always cut your threshold. You can always set your thresholds and your statistical cutoffs differently. We could have used standard deviation instead of MAD, and that would have upped our gene number too high because to actually have a legit follow-up. So there's always that balance between like cost and reality. So we thought 500 to 700 genes, that would be a legitimate number to do the follow-up studies.
Cool. Thank you very much.
Sure.